Good morning, everyone, and welcome to a new seminar of the EBD Seminar series. Before I introduce our speaker today, I'm going to give a couple of reminders. Please, if you want to make any questions, write them in the chat. If you have problems accessing the chat, you can do it with your Gmail account. And also, if you need a certificate of attendance, you can write down your full name in the chat and then send an email to the EBD seminar series at ebd.csIC.es. And with that, I'm going to introduce our speaker today, Rafael Molina Venegas. He has a talent grant in the Department of Life Sciences at University of Alcalá, and his work looks at the interface between phylogenetics and human well-being. With that, Rafael, when you are ready, you can start your talk.
Okay, so thank you very much for the introductions. Before to start, I would like to provide a few highlights to better introduce myself. So I complete my PhD at the University of Seville. But before that, the truth is that I worked my first step at the Estación Biológica de Doñana, who is also my scientific house somehow. So I enjoy undergraduate scholarship with Dr. Xavier Pico, who I hope is also watching the presentation. And I also enjoy nice teaching at the University of Pablo de Olavide during my master. I remember teaching by Pedro Jordano, Alfredo Valido, Montse Vilá, and many others nice teachers. So this is very nice that Estación Biológica de Doñana is hosting my talk today. So after I complete my PhD, I moved to the University of Alcalá, where I continue my research, developing phylogenetic methods and working at the macroecological scale in Holarctic systems. And after that, I moved to University of Bern, where I continue my main research lines, and I also start to delve a little bit on the relationship between human well-being and phylogenetics and evolutionary history of plants. And after that, I return at the University of Alcalá, when I'm currently leading my project, which is completely focused on the connections between the evolutionary history of plants and human well-being. And today I'm going to talk about more this project. So first, I want to introduce the concept of phylogenetic diversity, which was developed by Dan Fate in 1992. And the metric is essentially an index of the evolutionary history that is encapsulated by a set of species, the minimum spanning path connecting a set of species in a phylogeny. So in the left phylogeny, we have 6 species, and the black branches are representing the evolutionary history they are encapsulating. On the right phylogeny, we have another 6 species encapsulating a different set of branches and actually a less amount of evolutionary history. So on the left, we have high evolutionary history, high encapsulation of evolutionary history. On the right phylogeny, we have low phylogenetic diversity, low evolutionary history. So the idea of developing this index was actually developing a means to get at high levels of feature diversity. So for example, on the left side, we have the situation with low phylogenetic diversity, and according to the framework, this will mean that we will be capturing on average lower phylogenetic diversity than on a situation as in the red side of the slide, when we have higher phylogenetic diversity, and in principle, we are expecting higher feature diversity. This framework of phylogenetic diversity was well received in the early 90s of the past century as a way to get out of the agony of choice, which was essentially a problem of what to conserve, the budget is limited for conservation and we have to make decisions. So in principle, if we preserve a species implying high phylogenetic diversity levels, we were preserving high feature diversity levels. So this was, and that was actually the idea of using phylogenetic diversity as a mean to preserve high levels of feature diversity or functional diversity. And nowadays, there is some conservation programs that are incorporating phylogenetic diversity. For example, the Age of the Existence programs is aimed at protecting some of the most evolutionary distinctive and endangered species on the planet, with a particular focus on vertebrates. So for example, if we have two endangered species, equally endangered species, and this program will put more emphasis on the one implying more evolutionary history, the more singular one, because that species will represent more unique functional diversity. Okay, so the idea was preserving these species which are evolutionary distinct. Just an example. Today I'm going to talk also about ecosystem services, which are all the benefits that are provided for free for our well-being and from nature. So the concept was long time recognized. So for example, the Greek philosopher Plato already realized that too aggressive logging would eventually lead to soil loss, which could have a strong impact on the yield of the crops. The idea of ecosystem services has been orbitating the thinking of humans, and perhaps was the economy Schumacher in the 20th century, the one who first coined first coined in the concept of natural asset. He was the one who conceived nature as a set of natural benefit that could be actually quantified and that could have an impact on how we manage, on how we deal with the economy, the world economy. Later on, other researchers such as Robert Constanza further developed the field of ecological economics. And more recently, the Millennium Ecosystem Assessment just defined or established 4 different categories of ecosystem services, which has been used in quantitative ecological analysis, as the one you can see at the bottom of the slides. Well, it's important to know that the Millennium Ecosystem Assessment distinguish between ecosystem services and biodiversity as 2 different things. They didn't include biodiversity as one extra ecosystem services, but just as a means to get at different services, such as, for example, pollination. So in this paper, in this review by Faith and collaborators, the authors state that biodiversity should be recognized as a provider of concrete benefits to humans, not just as a means to get at other ecosystem services. They are stressing that biodiversity is a value itself. And they went further. They coined the concept of ecosystem services rather than ecosystem as a means to highlight the full range of services that are produced from the evolutionary process. For example, plants providing medicines or food. And they also stress that phylogenetic diversity, the metric of phylogenetic diversity, could be used as a way to quantify these ecosystems. So the idea is that the greater the evolutionary history represented by a set of taxa, the more current and future ecosystem services can be provided, because phylogenetic diversity, as I said earlier, would in principle be a valuable means to maximize the amount of feature diversity that we are capturing without knowing actually what are the functional traits we are dealing with. So this is the theoretical framework, which actually has little experimental support. But there is still some studies that were aimed to provide this empirical support for the theory. So, for example, we have the study by Florian Meisel and collaborators, and they developed a nice conceptual framework in order to test the value of phylogenetic diversity as a way to get at high feature or functional diversity. So in the x-axis of the graphic, we have the portion of a species sample from a given pool of species. And in the y-axis, we have the amount of functional diversity we are capturing with that amount of a species sample. So the dot sin line is representing the relationship between random sampling of a species and functional diversity. The thick dotted line, in contrast, is representing the amount of phylogenetic diversity that we will be capturing if we select a species using a phylogenetic information system. For example, using phylogenetic diversity, maximizing the amount of phylogenetic diversity encapsulated by our set of species. And the black line at the top of the figure is representing the maximum amount of functional diversity that we can capture given a set of species, a portion of the species in the pool. This is the conceptual model they use, and they test the theory using 3 different taxonomic groups: birds, mammals, and fishes. And they quantify functional diversity using 4 traits as body size and foreign activity, etc. And they conclude the following. They found that using maximum levels of phylogenetic diversity in this global study will result in an average gain of 18% of the functional diversity relative to a random choice. This is good, but the bad news is that 1/3 of the comparison show functional diversity levels lower than those expected under random choice. So they conclude that prioritizing phylogenetic diversity could be a risky strategy in conservation planning. So not really a good support for the theory. This paper was replied by Owen and collaborators because they argued that the authors were adopting a rather narrow functional perspective. They were working with 4 functional traits, which is less than the PD framework can actually capture. So they essentially argue that functional diversity based in 4 traits is not, do not equate to feature diversity. Meisel and collaborators also replied to this reply, just stating that actually theory is something foundational to science, and we need the empirical evidence. So they recognize that they are working in just some aspect of feature diversity, but still the evidence is to be required. So to my judgment, both group of authors were somewhat correct, because it is true that Meisel and collaborators just work on a few traits to compute functional diversity. And on the other hand, the truth is that the PD framework is theoretically well found, but the empirical evidence supporting the theory is still tenuous. And approximately 1 year after this debate occurred, this review came into biological reviews, and the authors actually reviewed the evidence connecting evolutionary history and human well-being. And they conclude that after 25 years of academic research, the empirical evidence, and as Meisel and collaborator was already questioning, is still very limited, it's still very tenuous. So we need more empirical evidence. And here is when myself and my group and my collaborators came into action. So the idea of this project, of my current project, was to quantify or to evaluate whether phylogenetic diversity is efficiently capturing services for humankind. And we focus on plants. We focus on plants because, to me, or to my knowledge, plants and the plant uses is the more palpable evidence of the services that are directly provided by biodiversity. So this was the idea of the project, providing this empirical evidence for the theory, which is largely missing. And for this purpose, we focus on plant services. To do so, we built an extensive data set, a global review of plant use record using the Marberly Plant Book. So we review up to 13,489 genera, accepted genera of vascular plants. And for each of these genera, the author David Marberly has been collecting plant uses from the 70s up to the 2017. So almost 40 years of collection of plant uses from the literature by reviewing up to 1,000 journals, monographs, flora, etc. So it's a quite impressive work. So after reviewing all the plant uses that are recorded in the Marberly's Plant Book, we obtained 9,478 records across 28 standard categories of use. To establish these categories, we follow the guidelines in the Economic Botany Data Collection Standard by Cook, which is essentially that, a guidelines to compile information of plant uses. Sorry. And we consider either realized and mooted uses as long as they were well documented in the literature. And we have the 85 categories for use, which are clumped in 7 groups of categories, pretty heterogeneous groups of categories. So now I will go on a quick tour around this well of services just to show them, to take a closer look to them. So firstly, we have 4 categories related to human and animal nutrition, such as food plants, food additives, food for the animals, for livestock, and also food for invertebrates. So these are the 4 different categories for human and animal nutrition. We also have fuels. We have firewood, charcoal as a different category of fuel, and also natural fuels of biological origin, others than fuelwood and charcoal. Materials is another group, including many different categories of uses, and we can distinguish between what I call macroscopical materials, such as timber, stems, for example, as chewing sticks, also as directly building materials. We have fiber plants, also a very important category of use. We have fruit and seeds as a macroscopical material. And that's it. That's concerning the macroscopical materials. We also have materials of natural origin. So, for example, dyes. So, plants as a source of dyes, plants as a source of resins and gums, same category. Plants as a source of oil, also waxes, scent, and rubber. We have the category of medicine, which is perhaps the more popular, the one that pop up into our minds when we think on plant uses, but we have many different uses besides medicine plants. Poisons. We also have poisons, either for vertebrates poisons, for example, the famous curare poison of the indigenous people that used to, that was very efficient just for hunting, just to just for hunting with arrows, the arrow poisons. And we have also the invertebrate food poisons. So for example, many different types of insecticides. We have also social uses. So for example, we have abortifacients as a category of uses, either for male or for female. For male would not be abortifacient, but actually anti-fertility agents, if you want. We have a category of stimulant plants, drugs used for recreational purpose or ritual purpose, or simply for stimulation, as caffeine, for example. And we have a category of plants that we call magic symbols and inspiration. Plants that were used as a source of inspiration and for their symbolic value. So you can think, for example, in the florography, which was a cryptological system for communication in the Victorian society of the 19th century in the UK, also in the States, where people use different combination of flowers or different species of flowers to convey feelings and also secret messages. So just as an example of plants as a source of symbols or inspiration. And finally, we have the environmental uses. So we have plants as ornamental that are valuable for their ornamental value. We have plants as bioindicators and bioremediators. We have another category of plants that we call soils improvers. So plants that are used to improve the quality of and the fertility of soils rather than to repair the soils, which will be more bioremediator, the former category, the category described just earlier. And finally, we have shelter and hedge plants, plants that we use just to separate different areas of a field, for example, also shade trees, plants that are used to get protections. So we found that 33% of the genera show at least one category of use, and the number of uses per genera was between 1 use and 17 uses, the maximum number of categories. So most of the genera has just 1 or 2 uses, or even none. Actually, most of the genera didn't show any documented use according to the plant book. And yes, we have a long tail of genera, including more than 2 or 3 uses. So the most common use was the ornamental, 26% of the records, then the medicinal plants. On the third place, we have human food, 13%, then timber, and finally, all the other uses showed less than 5% of records. That's concerning the data set. So on the other hand, we need the phylogeny. So for this purpose, we use the phylogeny by Smith and Brown, recently published in American Journal of Botany. So this phylogeny includes 70% of all accepted genera according to the plant book. So we have molecular information for 70% of the genera, but still missing 30% of genera without molecular information. So in order to include them in the phylogeny, we follow a systematic approach to account for this uncertainty. So rather than working with one single phylogeny, we work with a distribution of possible trees. This is rather than being a mean to get at the true phylogeny, the idea of working with a distribution of trees is just being aware of what is the amount of uncertainty which is affecting our results. So if we use 100 trees and the confidence interval of our metric is narrow, then we can be certain that this phylogenetic uncertainty in the missing genera in the phylogeny are not really affecting our result. That was the underlying idea of using this systematic procedure just to randomly stick the species. I will not go into the details for this. So Concerning the hypothesis, the hypothesis is that maximum level of phylogenetic diversity captures more human benefit than random selection of taxa. If this is true, so phylogenetic diversity will capture more richness of plant categories, up to 28, and a minimum of 0 categories, more plant use record in total and per category, and also more equitable distribution of record across the categories. So we use the greedy algorithm to obtain different subsets of taxa that maximize phylogenetic diversity. We use 10%, 20%, 30%, and up to 90% of the taxa in the pool. And because, as you may wonder, there is many different subsets of taxa in a phylogeny that can maximize phylogenetic diversity for, let's say, 10% of the pool. So we actually compute 10 different subsets of maximum phylogenetic diversity taxa, if you want, per phylogeny, and we were using 100 phylogeny. So in total, we can calculate 1,000 phylogenetic diversity maximum subsets per category of sample size. So for each of these maximum phylogenetic diversity subset, we calculate the richness of categories that they capture, the number of plant use record, and the equitativity using the Pillow index. And we average the results, these variables across the distribution of 1,000 category per sample size. And in order to get some statistical significance of the contrast, we use standardized FXI scores using new distribution of randomly sampled taxa. So subset of taxa of the same size as in the maximum phylogenetic diversity subsets, but in this case, randomly selected, not using phylogenetic information. And this is what we found. So on the right side, you have the conceptual model of Maisel and collaborators that I described earlier. And on the left side, we have the results. So for each sample size in orange color, we have the portion of plant use records in total, pulling all categories of use together, selected that were captured, after using random selection of taxa. And in blue color, you have the portion of records using phylogenetic diversity. So as you can see, phylogenetic diversity efficiently capture more record than random selection of taxa. So, and the shape of the figure somehow is mirroring the area in under the thick dotted line on the red side. So the area labeled with B, which is the amount of records that we are gaining thanks to the use of the phylogenetic diversity approach. And contrary to Maisel and collaborators, we found that 92% of the individual comparison result in positive gains. So for example, here I'm showing the gain per use category for the 20% portion of the pool. So as you can see, we have positive gains and significant gains for most of the uses we use in the analysis, except for 2 of them. On the one hand, we have they use animal food, for which phylogenetic diversity was not an efficient means to get many different numbers of records. And also we have rubber plants, which is notably the worst category, the worst category for which phylogenetic diversity works. So we actually, phylogenetic diversity get a less number of rubber plant than did a random selection of taxa. But for all other categories, the gains were significant and positive, as you can see in the figure. And even further, because functional phylogenetic diversity got at higher equitativity in the distribution of records compared to random selection of taxa. as you can see in this bar plot. So it is not only that we are getting more records, we are getting more equal distribution of record across the different categories. Okay, so you may wonder, plant use records at the genus level, what that mean? Is that something valuable? Well, I would say yes, for different reasons. So on the one hand, species level records are vastly underdocumented below the genus level. We have a huge gap of knowledge, of the documentation knowledge of the plant use records. So the most elemental knowledge remains unknown concerning plant uses at the species level. And we are also far from a global species level phylogeny, of course. And in any case, I think this genus level approach still makes sense from an evolutionary perspective, because at the end, genus is just a different lineage at a different genus. So from an evolutionary perspective, it makes sense. But it is true that the unit of conservation is normally the species rather than the genus. The operational unit of plant use is also the species rather than the genus. So we have some uncertainty here. So it wouldn't be the same getting at a genus in the phylogeny, for example, a genus that is useful as timber. It would be more valuable if this genus had one single species than if the genus has, for example, 100 species, because we don't know which of these 100 species is the one that is providing timber. Probably most of them are, but we don't know. We only know that the genus is a timber genus. At least one of the species in the genus is providing timber. So we have a taxonomic uncertainty here, if you want. And also, you may wonder if this pattern is consistent across floras in different biogeographic realms. For example, North America, South Africa, Southern America, Europe, etc. So we did a set of additional analysis just to test this idea. In order to take into account this uncertainty of the species that are actually used, what we did is just downweighting the plant use matrix, which is a binary matrix, genera and categories, 0 or 1. So we just downweighted this matrix according to the species richness in each genera. So the higher the number of species in the genera, the lower the amount of future diversity that we will be adding to our pool. And we repeat the analysis using this downweighting matrix, and we found that the pattern persisted, which was very nice. And concerning the analysis across biogeographic regions, we still found that the pattern persisted across regions. So on the left side, we have the analysis using the 0 present absent matrix of uses, not downweighting by the species richness. On the right side, we have the results after downweighting the matrix by the species richness. So for Africa, the pattern persisted, also for Europe and Asia, which were clumped into one single region, also for Australia, also for North America, tropical Asia, and South America, the pattern persisted. But if we are analyzing the data, just including the genera that are present in each region, it is possible that a genera is, for example, is a source of medicine, maybe in South America, but it is not in North America, because the species that are in North America maybe are not medicinal plant, despite they are in the same genus. We don't know this. So in order to test if there is an effect in this bioregion uncertainty or geographical uncertainty, if you want, we did a final set of analysis. We conduct a final set of analysis, including only the endemic species of each bioregion. And the good news is that the pattern persisted again. Actually, a bit less significant in some cases, as you can see, for example, for Africa, quite significant for Europe and Asia, less significant for Australia, but still the pattern persisted using the endemic genera of Australia for the analysis. Also North America, tropical Asia, and South America. Well, now the question is, what would explain this pattern? Why phylogenetic diversity was successfully captured this higher number of records compared to random selection? Well, if we plot the phylogenetic diversity of the categories, so the amount of evolutionary history connecting all the genera that provide a certain use and the relative gains for that category, we find a rather positive and significant relationship. So these analyses are showing that plant use records, or we can expect more records for those categories of use that are scattered throughout the phylogeny, the entire phylogeny. So phylogenetic dispersion in these plant use categories is largely driving the success of the phylogenetic diversity approach. So just to conclude this, we found that phylogenetic diversity, or we confirmed that phylogenetic diversity is a valuable means to evince to capture significant levels of plant services, both globally and across the main continental regions of the world. This study, which I didn't mention, that was recently accepted in Nature, Ecology and Evolution, is a contribution to the empirical foundation, confirming a long-standing theoretical background, and hopefully it will help future discussions to promote better grounded accounts of the services that are provided by biodiversity. And is phylogenetic diversity a way to capture option values for the future? This is uses or benefits that are yet to be discovered, and that maybe we can need in the future, but not now. Well, although the theoretical framework says that yes, phylogenetic diversity will be a means to do that, to me, this is a really big question. But the truth is that if we consider, for example, people in the Neolithic, for the people in the Neolithic, phylogenetic diversity was actually a means to get at the services, the plant services that were to be discovered until today. So encompassing the whole history of humans. Okay, some for future directions. Maybe these useful plants are not always widespread in the phylogeny. Maybe there are some lineages that are providing a significant number of services. And this is important. This is very important. Detecting these lineages could be very important. For example, in the case of medicinal plant, these phylogenetic domains of plant uses could be helpful for bioprospection. This is the discovery of future drugs that could be helpful in the future. Also, I would like to analyze cross-cultural patterns of plant use, which have been coined evolutionary convergence. So for example, concerning the mahogany tree, the mahogany wood, which is a precious wood, very valuable wood, that is actually provided by Swietenia, which is a Southern American genus in the Meliaceae. But we also have a mahogany tree in Africa, the Kaya tree, the genus Kaya, which is also in the Meliaceae. So the idea is that the Meliaceae is actually a lineage providing wood food, wood of very good quality, and this source of wood of very high quality wood was discovered independently by different cultures, ancient cultures in Africa and South America. Also, we can wonder if there is lineages providing multiple different uses, a diversity of uses, multifunctional lineages. We actually don't know much about all these questions. So I have the opportunity during my former PhD, my former postdoc, not PhD, just to delve a little bit on this question, analyzing the ethnoflora of Mount Kilimanjaro for different groups of, for different categories of uses. So for here, I'm showing what I call hot clades in terms of plant uses. So phylogenetic lineages providing a significant number of species being useful for something, in this case for animal food, and we found the a lineage within the Fabaceae, the Favoideae tree, and also the Poaceae, which are probably two different domains of animal food that applies worldwide. And also the eupolyplot, which is something a bit weird and maybe something particular. They use this fern to feed the livestock, despite ferns are considered of low nutritional value. Also, concerning plant use of building materials, we have some domains, for example, the Acacia mass albicea clades, also the Sapindales, family Meliaceae, for example. Similar phylogenetic domains for fuelwood, Acacia and Sapindales, so actually they are the same species providing the same services. Concerning medicinal plants, we found different domains of uses, including the Acacia mass albicea again. And concerning ornamental plants, again, Acacia mass albicea and some other domains as the asparagales. I will talk in a few moments. So the Acacia clade is a multifunctional lineage in Africa. It's a very important clade in Africa, comprising Acacia plus albicea. It's actually a polyphyletic, they are two polyphyletic in general. So the Acacia tree appears in many ancient representation of plants in Africa. So for example, the plant is a candidate as the source of wood for the Ark of the Covenant. You can see in this picture, medieval picture. The plant is a source of Arabic gum, so medicinal plants. And ancient Egyptians already used this plant to build their ships. It was a popular timber plant for building ships, good wood for ship. So a very important clade in Africa. And before to wrap up, I would like to grab your attention to the monocot in the Kilimanjaro. So on the one hand, we have the Poaceae as an animal food clade, and the Asparagales clades as an ornamental domain. In the case of the Kilimanjaro ethnoflora, is actually not ornamental but shade plants, because ornamental plants are not very in use in the Kilimanjaro. So there we have the genus Dracaena, which is an important genus with an important symbolic value in the Kilimanjaro and the Eastern Africa in general. And of course, we have in the African islands of Canary Islands, the Draco tree, which is an important appealing for tourists. So, and this local village, Icod de los Vinos, is actually benefiting because of this Draco tree, which is perhaps the best exponent of his kind. But if we think in the global scale, actually within the monocot we have on the one hand the Poaceae, the grasses essentially, but we have the Asparagales as a clade that have developed very eye-catching flowers, which has a strong ornamental value elsewhere. And this is essentially because these two different clades of plants co-evolve with different groups of animals. So Poaceae co-evolve with big animals, and families and species in families in the Asparagales clade did with insect pollinators. So, and as a result of this evolution, they provide different services nowadays. Animal food in the Poaceae, and animal pollination, ornamental uses in the Asparagales. So, Peter Linder defined what they call the Viking syndrome of Poaceae, because grasses had a strong, high dispersal capability. They go everywhere, just as Vikings did, from Scandinavia to the Mediterranean, even to the New World, according to some theories. Once they settle, they they are the best at excluding competitors, so they are good warriors. They, for example, they are highly flammable, so they produce fires so that they exclude potential competitors, and they are very good in growing in places where animals are browsing and others can't drift, other plants can't drift, but they do. And then they continue dispersing. So we can think in this, in the monocots, as a good, nice example of the early evolution of plant services, of ecosystem services, which is provide for plants as animal foods concerning the grace, the poaceae, and as ornamental plants for the asparagales. And just to finish, I would like to thank the people who have been involved in this research, Professor David Maberly, Professor Miguel Angel Rodriguez, who has been my mentor in Alcalá, also Professor Manuel Pardo from the Universidad Autónoma de Madrid, Cristina Ronquillo, my technician. She did a very, very nice job and she saved my life. She is hero. And also other people that are also involved in the project, Miguel Verdú, Aurelio Malo, Juan Cantalapiedra, and Juan Carlos Moreno Saiz. Thank you very much for your attention.
Thank you, Rafa. It was a super nice talk. Thank you so much. So you have many questions in the chat, and if someone else wants to ask a question, there is still time for that. Well, I'm gonna first start with the question from August, and he said, "Thank you so much, a very informative talk, Rafa. For that, unfortunately, prioritizing conservation areas is a tragic dilemma that we sometimes need to face off. So, if dealing with that dilemma, to choose areas to preserve, would you either prioritize phylogenetic, functional, or species diversity?" I don't know if you understand.
So essentially, if I understood correctly, the question is whether I should preserve functional diversity instead of species richness, for example?
Yeah, or phylogenetic diversity. So which kind of diversity do you think you should prioritize?
Yeah,
prioritize.
Well, I think we should get to a trade-off here. So it wouldn't have any sense to preserve 3 species, even if they provide maximum functional diversity. So I would prefer preserving 100 species, even if they provide a lesser amount of functional diversity or feature diversity in general. So
it will be the species richness at the end.
See, so exactly. So at the end, we have to keep in mind that preserving a high number of species is also very important, not only focusing on functional or phylogenetic diversity, which are blind metrics to species richness. At the end, species is something unique somehow. So we have to consider species richness in conservation planning, definitely.
Okay, so it's a difficult question anyway, because also you have keystone species and so on. Yeah, but okay. So there's another question by Cayetano Gutierrez Canovas. He says, "Great talk, Rafael. Thanks a lot. I enjoy and learn a lot." And he has 2 questions. The first one is, according to your empirical experience, to which degree the relationship between phylogenetic and functional diversity is influenced by functional trait choice?
Functional trait choice. So
I think he's asking about the relationship between phylogenetic diversity and also functional diversity. That is also something I was asking for.
Yeah, so, well, I think in this case, we just focus on plant uses. So we are not actually quantifying functional diversity, just number of uses per category and pulling all category of use together. But the problem with functional diversity is that there are as many functional diversities as researchers in the world, because functional diversity could be a combination of plant height, seed mass, and specific leaf area, the classical Westovi triad of traits, functional diversity could be root biomass, for example, plus lateral spread capacity. That could be a different way of computing functional diversity, and so on, so forth. So, and this is the problem with functional diversity, that when working with this metric, we have to be sure that our composition of functional diversity is well aligned with the question we want to address. So in some cases, functional diversity will be very well aligned with phylogenetic diversity, or in some other cases not. Nevertheless, it is, to me, it is pretty obvious that on average, long and distant relatives are less similar than close relatives. And that's the reason why we are more similar to monkeys and less similar to lemurs, and even less similar to spiders, and even less similar to plants, and so on and so forth. Because on average, phylogenetic diversity is informing of feature diversity, but just on average, depending on the traits we are interested in, the relationship could be strong or could be weak. That's for sure.
Okay, I don't know if Gabriel wants to ask more. Cayetano wants to ask more. And his second question is, when using the phylogenetic diversity as a surrogate of ecosystem services or functions, what will be the ecological mechanism driving these relationships?
Yeah, that's a very good question. So in the paper, we speculate a little bit on this matter. So on the one hand, it could be that because there is a theory that say that humans use the first thing they have at hand. They don't complicate their life just when it is about to get to solve a problem. If this species is suitable to solving this problem, I will keep it, and I will not try to find a different solution, even if the potential solution could be other species can provide a more optimal solution. And if we just take a plant to solve a problem, we will probably not take a close relative to solve a different problem, because close relative will probably provide the same services. So, and the problems of humans are multifunctional. We have to feed, we have to heal, we need to clothing, we need many different things. So that could be a reason why. Another reason, which had been documented at the species level and local scale, could be cultural. Some different cultures somehow try to differentiate from each other using different set of species. And there is a very nice paper by Professor Manuel Pardo, who is a co-author of this research. The research was conducted in the Basque Country, and they found that different cultures that cohabit in the same region, so sharing the same flora, use completely different plants, despite they share the same flora, probably because they are trying to differentiate from each other. So these two different factors could be, at the end, leading to a phylogenetically over-dispersed flora. And also, for sure, we are considering a global scale, and plant lineages are geographically constrained to some extent. So we have Gondwanic lineages, we have North Temperate lineages, and the cultures that prospect those lineages are also geographically constrained. That is also a reason that would explain that the uses of the plant are widespread throughout the whole plant of phylogeny of plants. But also we found that the pattern is persisted within biographic regions. So this third factor could be also at stake, but there is something more. And I would like to deal more in the future about this question, because it's very, very interesting. What are the actual drivers of the pattern?
Okay, thank you. There are many questions. I'm gonna, because Cayetano was doing a clarification of what he was referring to trait choice. He was referring to the number of traits he wanted to include, the number and the type of traits. So that was the clarification he did about but he is happy with your response anyway.
Okay.
So the next attendant is Nacho Artemis. He says that is a very really cool talk, and he's asking about the scale of your work because he says, I wonder though if the scale of the analysis is the most relevant. My interpretation is that phylogenetic diversity is a proxy of ecological communities functioning, but your analysis is not done at the community level. Will using observed communities change the answer? Or in other words, are your results applicable at a smaller scales?
Well, this is also a good question. So I know at least of 2 case studies conducted at a more local scale where the pattern was observed. For example, Forrest and collaborators in 2007 found that the ethnoflora of South Africa, for the ethnoflora of South Africa, phylogenetic diversity or maximum levels of phylogenetic diversity will also get at a higher amount of benefit than random selection of taxa. And for the Kilimanjaro, we also found that the pattern persists or is persist. So we also found that maximum level of phylogenetic diversity for the flora of the Kilimanjaro mountain actually get at more benefits. Whether the pattern will persist at even lower scales, so for example, the community scale, probably not. I think this is more a general picture, a general question. At the community scale, probably, because how many useful species we will have in a community? You know, in order to this framework to make sense, I think we need to work at least at the regional level, because otherwise, probably the number of useful species will be just a few of them. I'm not sure if I'm resolving the question, but the answer is yes. Probably, almost certainly, there is an effect of scale, and in decreasing the scale, we will probably miss the signal. So, for example, in Australia, considering only the endemic species of Australia, the pattern was still there, but less significant, probably because it's a reduced, a lesser, smaller flora compared to Africa or South America, for example, with less uses than in the South America or Africa, which are hotspots of economic.
Yeah. I guess that a validation of these kind of questions is to include any kind of weight for the area size. I don't know if I mean, you are already using the sample size, but and I mean that somehow when you are doing this biogeographical separate analysis, you were also repeating the same pattern. So that means that the shorter scales are also working, but maybe you could include also some kind of weight according to the sample size or to the area size, sorry. And this would be to see the effect of the scale for the pattern, I guess.
Yeah.
This is an idea.
Yeah, this could be a possibility, definitely, yeah.
There is another question by Ruben Bernardo. He says, thank you so much for the talk. It is very interesting. It is a very interesting topic, and you have worked very hard. So congratulations for the work and for the publication. I wanted to ask 2 questions. Sorry if you have commented on the talk and I have not heard well. The first one is, would it be possible to use a null model that tries to better reflect the current conservation criteria to better see the contribution of this approach? And then I will ask you, do you want to know the second question already?
Well, I maybe I can answer to the first question, then we can jump to the second one. So yes, definitely we could use different new models that could be more realistic than random selection of taxa. Definitely, for example, using current techniques or conservation planning approaches to these conservation approaches as a null model. And also, I also would like to test not just maximum level of phylogenetic diversity, but perhaps intermediate levels of phylogenetic diversity. So in selecting a species, not only on the base of maximum phylogenetic diversity, but perhaps 90% of maximum diversity, 80%, it could be possible to know at which point phylogenetic diversity is actually working. So yeah, there is a interesting avenue of exploration still to be explored, and I would like to jump into that definitely in the future.
Okay, great. And the second question is, do you have an idea of a possible correlation between the functionality of plants for humans and the one they may have in nature?
And the sorry.
And the function that plants have in nature.
Functions in terms of plant biomass and these kind of things.
So I'm not sure, Ruben, maybe Ruben can write his clarification. I guess he's referring about human welfare and plant functions, but I'm not sure about that.
Well, so for example, a function could be the degree of resistance against herbivores, for example. So actually, the use of plants as invertebrate poisons is due to the evolution of this defense, chemical defense against herbivory. So that could be a connection between the function and the use of the plant. And I also, as I tried to remark by the end of the talk, grasses is a clade of the phylogeny, is a group of plants which are very good at living in place where there is plenty of herbivores. So they are food. They serve as food for wildlife, for example, in the Serengeti. And this is because they are very good at doing that. And now today we use these plants just for feed our livestock because they are useful for that, no? Because they way of growing, just avoiding of keeping the weak parts of the plants protected so they can resprout every time. So, and that could be also another connection between function and the services, and also orchids plants, which are very valuable for in floristery, for example, or in gardens and in ornamental botany. This is not a chance. This is because these plants actually co-evolve with the insect during millions of years of co-evolution. And after all that time, they aim at developing these very nice big flowers that we can see today. So it's also a connection between the function and the use of the plant. So yes, there is evident connection between the function, the benefit of the plant, and the function of the plant in nature. They should be.
Yeah, I think you did well. I mean, you did a great answer. Ruben is also like rephrasing his answer. Say, I'm referring to the role that the species can play in natural communities. But I think you already answered very well what he was asking for. And there is another question by Monse. Monse Villar, she says, is any possibility to sort out differences between invasive and native species with your native species uses with your dataset? And that will be great for the IPBES report. So maybe you could also differentiate if the species are native or, but I guess that depends where you are talking, in which area you're based.
Yeah, we did. I mean, for the biogeographic constraint analysis, we only consider native genera according to Plan of the World Online. So in that repository, you can see for every genera the distribution worldwide and whether a record is it can be considered a native record, native plant, or an alien record, a record due to an invasion. So for this analysis, we only consider native records for each of the bioregion. I don't know if that resolved the question, because we are only using the geographical framework for this second set of analysis, because the analysis we did, the main analysis are worldwide. So we are using the complete vascular flora.
So I don't know if she's answering something else. No, I don't think. I mean, she says, what about phylogenetic diversity signals or on these services?
Well, we found that in most cases, the plant use categories tend to be over dispersed. That's the general pattern. We also found some clustering in certain cases. And clustering and phylogenetic and over dispersion is a kind of phylogenetic signal. We didn't fit models of evolution, for example, using the statistic for binary traits, because that was not the scope of the analysis. But we did explore phylogenetic diversity, and in some cases we found a strong clustering. For example, in rubber plants. Rubber plants or plants providing rubber are constrained to a very, very specific clade of the phylogeny. I still don't know, because I didn't explore this. It's not a direct, straightforward, because the phylogeny is huge, so it's not easy just to show these hot clades of the phylogeny in terms of use of plants. But yes, there is some phylogenetic structure in certain cases. For example, in rubber plants and also in food plants. Most of them belong to the Fabaceae and the Poaceae. These are two popular families worldwide for feeding livestock.
People. Yeah. So there is another question by, I think that's all. So all of them are saying you thank you so much for the answers. If you want still to do any other questions, we still have a few minutes for that. And I mean, it's a very, very small point, but at the beginning of your talk, you said that you were using different trees, different phylogenetic trees for the species that you didn't have all the phylogeny.
Yeah.
And that you were, I mean, and you said we are including different phylogenetic uncertainty in the analysis, but I'm not sure how did you do that.
Well, so we have in the global phylogeny that we use, the Smith and Brown phylogeny, we have 71 actually % of the genera. So 70% of the genera include phylogenetic molecular information. They are already in the phylogeny, but we are still missing 30% of them. I don't know, 3,000 something. So for each of these genera, what we did is retrieving the infra-family taxonomic category. So for example, this genera belongs to the tribe or the subfamily A within the family Fabaceae. Okay, so then we went to the phylogeny in order to see if this infra-family category is actually monophyletic. If it was monophyletic, we take advantage of this taxonomic information in order to randomize the species in the crowd node representing that taxonomic category. Because we are almost certainly, given this phylogenetic evidence, that the phylogenetic uncertain taxa, the PUT, which is called the PUT, will be somewhere within that clade and no anywhere. So, and we did that for every single genera, just seek for the monophyletic nature of infra-family taxonomic categories, such as subfamily or tribe, for example, or whatever. And we test for the monophily of these taxonomic categories. And using this information, we randomize the species 100 times in order to get 100 phylogeny. That was the idea. It was a combination between taxonomic and phylogenetic evidence. And this is very well described in a paper by Tiago Rangel and collaborators in 2015 in Evolution. how to take into account phylogenetic uncertainty if you are interested. And we essentially follow that pipeline.
Okay, great. So I don't think there's any more questions, but so thank you so much again, Rafael. It was great. I'm looking forward to reading your new paper in Nature, Ecology and Evolution.
Thank you to you for inviting me.
You're welcome. It was a pleasure. Bye.
Bye-bye.